Hello, this is Dr. Rina Rani Ray. I'll be discussing on structure of bacteria, and today this is part 1. Now, what is bacteria? Bacteria are microscopic single-celled organisms that exist in their millions in every environment, both inside and outside other organisms. The ancestors of modern bacteria were unicellular microorganisms that were the first forms of life to appear on Earth about 4 billion years ago. Bacteria are ubiquitous in nature, as they can be found everywhere in the environment, such as air, soil, and water. Even extreme environments like hydrothermal vents, deserts, pole everywhere, and even within our body can be the home of bacteria. Some bacteria are harmful, but most serve a useful purpose. They support many forms of life, both plant and animal, and they are used in industrial and medicinal processes. Here we can see some of the examples of bacteria and bacterial culture. Now, the average size of a bacteria lies between 0.5 to 2 micrometer in diameter, surface area about 12 square meter, and volume is 4 micrometer. And the ratio of surface area to volume is 3:1. So if we can compare the sizes of atoms, small molecules, lipids, proteins, bacteriophages, or organelles like mitochondria and eukaryotic cell, we can see where lies the bacteria. And we know here it is electronic microscopically it can be seen. Now, regarding the size of bacteria, Microplasma genitalium is a parasitic bacterium which lives in various organs like genitalia and respiratory tracts. Is thought to be the smallest known organism capable of independent growth and reproduction. The size is approximately 200 to 300 nanometer. On the other hand, Thiomargarita namibiensis is even visible to the naked eye, and they are the largest prokaryotic organisms yet known. The size is 0.1 to 0.3 millimeter wide, but some reach up to a size of 3/4 of a millimeter. Regarding the shapes of bacteria, we can see the coccus, the spherical or round-shaped bacteria. If they remain in a chain, that's a streptococcus, and a cluster, staphylococcus. Bacillus are the rod-shaped bacteria. The common example is lactobacillus, Bacillus megaterium, and so on. Coccobacillus means they remain between the coccus and bacillus. The common example is Haemophilus influenzae. Vibrio are the curved bacteria. Here we can see, and the common example we know, Vibrio cholerae. Spirillum, the name implies, is a spiral bacteria. The example is Helicobacter pylori. And spirochete, here we can see the spirochete structure. The common example is Treponema pallidum. And square-shaped bacteria, here we can see the square-shaped bacteria is Haloquadra species. So here we can see some bacillus and coccus. If the coccus remain in chain, they form streptococcus, and if they remain in cluster, they will form staphylococcus. Here we can see the cells of Vibrio cholerae and the cells of spirillum. Regarding the bacterial structure, we can divide the structures into 3 categories. First one, the capsule, followed by cell wall, then comes plasma membrane, and finally the cytoplasm. Regarding the locomotory organelles, the flagella, pili, and fimbrine, and the rest, the inclusions and spores. Here we can see the capsule, the outermost layer. It's followed by the cell wall, then comes plasma membrane, and inside it remains the cytoplasm. which contains nucleoid, because we know the bacteria are devoid of well-defined nucleus. We can see some bacteria, they are moving with their locomotory organelles. Now we are going to discuss about the locomotory organelles. So these are the three types of locomotory organelles. This whip-like structure is a flagellum. These are the pili, and the rest are the fimbrine. On the basis of the location and number of flagellum, bacteria can be divided into 4 types. First one is a monotrichous. What is monotrichous? Mono means single. That means if a bacterium contains one flagellum on only one side of it, it is called the monotrichous. The example is Vibrio cholerae. Here we can see a tuft of flagella remain present on only one side of the bacteria. It is called the lophotrichous. The common example is Pseudomonas fluorescens. If this tuft of flagella remain present on both the sides, it will called amphitrichous. The common example is Alcaligens faecalis. And if the flagella remain throughout the periphery of the bacterial cell, it is called the peritrichous. The common example is Escherichia coli. Now here we can see how the flagella is moving, and the flagellum is remain embedded on the cell wall and cell membrane. Here we can see the entire structure. So this is the filament of the flagellum which actually moves. Then it is followed by hook, and finally then this is rod. This part is embedded in the cell wall and cell membrane. So these are we can see the outer rings and inner rings and one rod, and the rest part is embedded within the cytoplasm. So in this figure we can see this is a flagellar filament followed by the hook which is moving, and entire movement is controlled by this ball bearing movement. So this is the bearing which is remaining present in the outer membrane. The rod part is remaining present in the peptidoglycan layer, and this is the stator and the rotor which actually moves and makes the flagellum moving. So here we can see how the stator and rotors are moving and the flagellum is getting out and they are moving. It is moving. The movement of bacterium is accomplished by the clockwise and counterclockwise movement of the flagellum. Here we can see how the bacterium is moving. Now the next part, that is the second type of locomotory organelle, that is a pili. It's a short protein appendages, smaller than flagella. They help the bacteria to adhere on the surface. Examples, so E. coli has numerous types of pili. This is K88, K99, A41, etc. One of the most important type is F. pilus, which is used for conjugation and exchange of genetic information. Here we can see how this conjugates come closure with the help of pili. They are forming the conjugation bridge, and it is responsible for flotation and for increasing the buoyancy also. So a bridge is being formed. The third one is a fimbrine. It is thinner and shorter than a flagellum. They range from 3 to 10 nanometers in diameter and can be as much as several micrometers long. Help to adhere to animal cells and some inanimate objects also. They possess adhesins which attach them to the substratum. So here we can see the flagellum, the pilus, and the fimbrine. And one of the primary mechanisms of virulence for E. coli, Bordetella pertussis, and Staphylococcus and Streptococcus, this fimbrine. Here we can see the bacterial cell is getting attached to the host cell with the help of the fimbrine. So here we can see the fimbrine and the bacterial cell. And with the help of this type of locomotory organelles, the bacteria can move. They can freely move. With that, we are coming to the end of this episode. So thank you for watching the video.